Okay, children, let me continue. I said just now, what is DNA? DNA is sugar molecule, deoxyribonucleic acid, which determines our parentage. So, this is otherwise known as hereditary material. That is double helical structure, I said. It is composed of sugar molecules as well as nitrogenous base. Now, nitrogenous we can divide it into purines and pyrimidine. Purines, adenine and guanine. Pyrimidine, cytosine and thymine. The first thing you have seen, complementary angles, guanine and cytosine means other one is adenine and thymine. Like that entire helical structure, double helical structure, you can see the same sugar, phosphates and molecules. This structure is formed by James Watt and Francis Crick. In short, we can say this is Watt and Crick model. Watt's and Crick model of chromosome. Clear? Now we have to see what are the things present. I said already. In between they are connected by hydrogen bonds. It may be double bond or triple bond. This is what? Bond. They are connected. Guanine and cytosine, they are connected by the hydrogen bonds. It may be double or triple. Then DNA replication. This DNA can be replicated. Replicated means what? Reproduced. So certain thing it goes and then it's reproduced. DNA replication is one of the basic process that occur within the cell. Once the DNA has become tired of or destroyed, other DNA can be formed automatically. That is known as what? DNA replication. So the DNA, it may be cut or joined. So further they are using the enzymes either to join the bit of DNAs or else to cut the DNA, either for cutting as well as for joining. We can use the enzymes. They are DNA polymerase enzymes. Enzyme, I said very beginning, remember, in our body we have two liquids. One is enzyme, other one is digestive syrups. So the enzymes, they are helpful for cutting off as well as to joining, cutting off the DNA fragments as well as joining the DNA fragments. So the cutted pieces are added by DNA, hormones as well as enzymes, digestion enzyme, DNA polymerase. This is for, suppose the DNA in the bits of, many bits, the bits can be joined by using the ligase. What's the ligase? DNA polymerase for joining, and for cut into the thing, that is molecular scissor, otherwise known as DNA polymerase and DNA ligase. They are used for joining the They are added with the help of, yes. Okay, so this can be added by using DNA polymerase and DNA ligase, and molecular scissor is there that for cutting the DNA fragments. Then significance of DNA, that's very important. What are the importance of DNA? Why we should need DNA? It is responsible for the transmission of hereditary information from one generation to another generation. Clear? That is, we got our parental characters. Our parents, they got their forefather, their parents. That is, this is because of, from the mango we are getting mango only, from the cat we are getting cat only. This is because of DNA. It contains information required for the formation of proteins. It controls the development process and its activities of living organism. Even I said the new baby has come out, either it's a human beings or animal, whatever it may be, the new organism may come out without any deformation. This is because of chromosome. Chromosomes are regularly divided. There should be no changes in our body structure. Suppose if it is abnormally divided, we may have extra growth or else less growth. This and all because of chromosome. Sex determination. Sex can be determined by not by the parentage, by the chromosome of the parentage. I said at the very beginning, X and Y. X means there are totally pair of chromosomes. Totally, in the human being, how many pair of chromosome? 23 pair of chromosome. In this 22 autosome, and one allosome which determines the sex. Then mutation. Sudden changes is known as what? Mutation. Next we have to go for mutation. Let me rub the board and I'll write here. Sudden changes happen in our body. For example, while birth, no, we are normal. After we are growing to certain time, there should be some changes in our body. This is known as what? Mutation. Not regular changes, sudden changes. So a baby becomes a newborn baby, it becomes somewhat developed, then it comes to the middle age, teenage, middle age, last young age, and finally die. This is different. This is normal. Apart from that, sudden changes, that is known as mutation. What is mutation? Mutation is a sudden changes. It was introduced by Hugo de Varis. The mutation was first introduced by Hugo de Varis. He saw the changes in certain kind of plant. So, on seeing the thing, he said that is mutation, and he gave definition for that. Mutation is an inheritable sudden change in the genetic material of an organism. Here in page number 269. Fourth line. What is mutation? Mutation is an inheritable sudden change, not by heritage, but not by hereditary. Suddenly it may be changed in the genetic material of an organism. What is genetic material? DNA. Sudden change in the DNA is known as mutation. They are further classified into two. This mutation can be classified into two, I said. One is chromosomal mutation. Then other one is chromosomal structure. Other one is chromosomal number. What is mutation? A sudden change in the genetic material chromosome is known as mutation. So there are two kinds of mutation. One is structural, there is a structure change in the chromosome, or else number of chromosome, either it may be increased or decreased. So this is the sudden change in the structure of chromosome. This is because of cell division, I said. Because of cell division, they take places of deletion, duplication, inversion, and translocation. Deletion, a particular part of the chromosome can be delete. Delete any deformity in our organs. Okay, duplication, repeated. That's why extra growth present in us. Inversion. So, இப்படி தான் டிவைட் ஆகணுங்கிறதுக்கு பதிலாக வேறு விதமாக டிவைட் ஆகிருக்கும். And then translocation, chromosome may be translocated. Change in the number of chromosome, I said already. They involve addition, deletion, the number of chromosome in the cell. It is known as ploidy. So, change in the number is known as ploidy. P-L-O-I-D. Change in the number is known as ploidy. So, there are euploidy and aneuploidy may be further divided into euploidy and aneuploidy. Euploidy and aneuploidy. Euploidy, it is the condition in which the individual bears more than usual number of diploid chromosome. Here, euploidy, more chromosome, more than 2 diploid. That is, 2 means what? Either one from father, other from mother. So, adu pola varumbodhu ungalku 2 diploid chromosomes. Okay. The condition is, if an individual has 3 haploid set of chromosome, the condition is called triploid. Generally, 2 da irukum. So, that is diploid. Sometimes, I said no, because of addition of chromosome, they may have 3 sets of chromosome, that is known as triploid. And it has 4, that is tetraploid. So, this may be number of chromosome. It determines the number of chromosome. Generally, it will be 2 only, diploid only. Suppose in some cases, 3 chromosome, that is triploid. and 4 tetraploidy. Clear? These are because of the sudden changes only. It will be because of the sudden changes we have extra growth or else thing. So triploid oyster, you know, that is ஸ்டிப்பின்னு சொல்கிறீங்க பாருங்கள், அதுலேருந்து நமக்கு முத்து கிடைக்கும், pearl oyster. This is triploid stage, this is diploid oyster. Triploid tree will be 3 chromosome will be. Clear? Aneuploidy, it is the loss or gain of 1 more chromosome. This is number of chromosomes are increased, euploidy. Then anemo, either it may be loss or gain or lose. So this may be monosomy, trisomy, nucleosomy. Monosomy only 1 is minus, trisomy 2, and the nucleosomy minus 2. Okay, because of aneuploidy, there may be some disease in the human beings. One is Down syndrome. Down syndrome. This condition was first identified by the doctor named Langdon Down. This is diploid, aneuploidy. Aneuploidy. Because of the aneuploidy, that is, the chromosome may be, it is the loss or gain of 1 or more chromosome in a set of chromosome. ரெண்டு தான் இருக்கும், ஒரு chromosome 2 arms will be joined together. No, suppose it is 3 types. This chromosomes may be 3 types, that is monosomy, trisomy and nucleosomy. So this aneuploidy, there are 3 kinds of chromosome: mono, tri and nucleo. Mono 2n minus 1, that is 2n means what? That is 2 numbers minus 1. Trisomy 2n plus 1. Monosomy na அந்த ரெண்டு haploid இருக்கு பாருங்க, diploid ஒன்று மைனஸ் பண்ணுறோம். Trisomy with the diploid you have to add 1 more chromosome. Nucleosomy 2n minus 2, that is no chromosome at all. In man's Down syndrome is one of the commonly known aneuploid condition. What is the disease? So this is first found by Langdon Down. It is a genetic condition in which there is an extra copy of chromosome. Which one that is a Down syndrome? How many chromosomes are there? Extra copy of chromosome. First one, what is it? Down syndrome due to extra copy of chromosome. Extra copy of chromosome. Okay. That is chromosome 21. It is an extra copy of chromosome 21. This is extra copy of chromosome found in certain human beings. That is known as what? Langdon. That is found by Langdon Down. Here, it is associated with the mental retardation, delayed development, behavioral problems, weak muscle tone, vision and hearing disability. So, mainly it is found in the children only. What are the symptoms for that? How can we find out whether the boy is suffered with Down syndrome or not? What are the changes? Mental retardation, that is, mind is not developed properly. Delayed development, even குழந்தை டெவலப் பண்ணால் கூட ரொம்ப லேட் ஆகும். For certain age, in the age, in the stage, அப்படின்னு we will say no, it won't be like that, it will be delayed. Behavioral problems, they are not behaving normally like us. Weak muscle tone, muscles will be very weak. Vision, hearing, everything will be weak only. These are the conditions seen in the children. Other diseases, one is Down syndrome, other one is gene or point of mutation. This is second defect. Gene mutation. This is mutation, sudden changes. Mutation is the changes occurring in the nucleotide sequence of a gene. Here, the changes occur in nucleotide, that is in the nucleus product. Nucleotide changes. Okay, and nucleotide changes. It involves in substitution, deletion, insertion or inversion. Substitution means what? Sending other one. அதாவது இவங்களுக்கு பதிலாக அவங்க வர்றது, இதுக்கு பதிலாக அது போகிறது. Deletion, removal. Insertion or inversion, we are joining in middle. This is because of single or more than one nitrogenous base. Gene alteration results the abnormal protein formation in an organism. Okay. So what are the things we come across so far? Then question and answers you have to see. I think we got the guides. So you have the clear idea of the thing. All the short answer, book back answers enough. In between, we will say, no, this is important question. So don't waste your time by learning all the things. The cut short of the lesson will be given in the month of January, I hope so. So we'll see that. So far, you learn only book back answers. Suppose in case they are reducing the lessons, we no need to concentrate that lesson. We'll take the other lesson. Anyway, we are completing all the lessons. Okay, children? Now, what are the things we come across? Let me recall the points what all I said. First we have seen in page number 261, what is genetics? Page number 261, genetics I said. The branch of biology that deals with the gene, genetic variation and heredity of living organism is called genetic. Next we have seen genetic, based on genetics we have seen heredity. It is a transmission of characters from one generation to next generation. Then there are two genetic factors, one is DNA, other one is RNA, but the human being DNA only. Mendel, he made the researches about the changes in the body, so he is known as father of genetics. Who is known as father of genetics? Mendel. So what plant he has taken for the research? Pisum plant, that is Pisum sativum pea plant. So how many plants he observed? Nearly 10,000 pea plants he has observed. Then how many triads he has taken? 7 pair of triads. What are the triads I said? Stem length, flower position, flower color, pod shape, pod color, seed shape and seed color. Okay, and we have given the reason. What's the reason for Mendel's success? Then he did the hybridization, that is 2 pair of hybrid. One is monohybridization and dihybridization. Based on the experiments, he formed the Mendel's law, law of dominance, law of segregation, law of independent assortment. And next we have seen in today's class we have seen the structure of DNA, chromosome, DNA structure and what is gene. So totally how many chromosomes in our body? 23 pair of chromosome. So based on the position of centromere, the chromosomes are connected by the centromere only. It has 2 arms. Both the arms are connected by centromere. The centromere, based on the position of centromere, it can divide into telocentric, acrocentric, submetacentric and metacentric. And based on the function of chromosome, it is divided into autosome and allosome. Autosome I say bodily, allosome reproductive cell. And we have seen the model of DNA. It is proposed by Watson and Crick. It is double helical structure. And DNA replication, it has the capacity of replication. And significance of DNA, it is one more question important. And sex determination because of the chromosome. I said male is X and Y, female X and X chromosome. And mutation, sudden changes is known as what? Mutation. Because mutation further we divide into euploidy and aneuploidy. There is no loss of aneuploidy, but euploidy. Because of aneuploidy, there may be some diseases. What are the diseases I said? One is Down syndrome. It occurs only in the children. Okay children, thank you.